Nicole Fernandez.
My name is Maynard Zerone.
And today we'll be doing the cellular respiration project, and we'll be doing the experiment with you guys. Okay. So before the experiment started, we already labeled the germinated seeds in this tube along with the centimeters up to 6. And that would be the non-germinated tube.
So now we're going to be adding 5 drops of distilled water into 2 of the weld plates. Now we're going to be adding sodium hydroxide into the same wells that we added the distilled water into, 5 drops.
Now we're gonna grab 2 cotton swabs and add the cotton swab into each plate just to saturate it. And we're gonna be adding 2 cotton swabs, 2 dry ones into each of the germinated and non-germinated, and add the saturated cotton swabs on top of these.
Now that we've placed the 2 saturated cotton balls on top of the dry cotton balls, Nicole's going to be adding 50 milliliters of distilled water into the beaker, which is half of the beaker full. And then she's going to be adding in 2 drops of food coloring and then swirling it to make sure that the food coloring has spread throughout all the water. Now that the beaker is halfway filled, she's going to be adding in the 2 drops of food coloring and swirling it. Good job, Nicole.
So now what Maynard's gonna go ahead and do, he's gonna grab a piece of tape, grab the germinated test tube, go ahead and do it up to halfway, right around here. It's 0.5 from the actual end of the beaker. And that's just gonna go ahead and sit there. And now I'm grabbing the non-germinated one and doing the same exact thing.
Put the tape through. Tape is not underwater. So now that we've taped the test tubes into the beaker, we're going to be taking pictures of it every 30 minutes and inserting those pictures into here. And then at the end, we're going to be inserting our graph and all our data combined together.
So now we're gonna go ahead and get started on part 2 of the experiment, and we're gonna be showing you guys that as well. So now we go ahead and we have a cup that is half full with water. It is 32 Celsius, and we're gonna go ahead and be adding the yeast and going ahead and gonna be stirring it off camera. So now that the yeast is added, we're gonna go ahead and be stirring it with a pipette.
So the Durham tubes were already placed in isopropyl alcohol to be sterilized, and they're right here. And all the different chemicals that we need to prepare in phenol red were already prepared, and they're in the test tubes, and they're labeled as such. So now we're going to be taking each of the respective test tubes labeled F, and we're going to be adding in the fructose into the red test tube, into the test tube labeled F, without spilling. So now after the fructose was added into there, we're gonna set the test tube aside and we're gonna do this for both test tubes labeled G and M. Now we're doing the one with glucose for the test tube labeled G. Without spilling.
So now we're going ahead and uncapping every single one of the tubes, and we're going to be going ahead and adding the Duram tubes upside down so that the bottom part is going ahead and being up while the opening is facing down.
So now this is what the yeast solution looks like after it's been mixed and been set to yeast. So now what we're gonna do is we're gonna add in 2 drops of the yeast solution into each one of the test tubes. Then after we add in the 2 drops, we're gonna close the test tubes and we're gonna place them in dark places. And over the course of the next 12 hours, we're gonna observe each test tube and the color change that it persists with.
We're also going to be adding pictures at the end of this, and we're going to go ahead and explain what happened and what our findings were. Thank you to the CURE grant for going ahead and giving us this opportunity to learning more about biology.
And thank you to Dr. Morata as well.
The experiment definitely showed us a lot about cellular respiration and metabolism. We can see that the procedures were set in the video, and if you were following along, you could go ahead and see that. We're just going to be explaining our discussion, going ahead and explaining the results, and also explaining the tables that we have here.
So through experiment 1, we can see that both germinated and non-germinated seeds, the distance had increased, although the germinated seeds had increased at a higher rate than that of the non-germinated seeds. At 180 minutes, or 3 hours, the germinated seeds had traveled the distance of about 1.3 centimeters. The non-germinated seeds had traveled a distance of about 0.7 centimeters. Discrepancies can be found in the data for non-germinated seeds at 150 minutes or 2 hours and 30 minutes. The distance for the non-germinated seeds had decreased as time progressed, which should not be the case. The fault in this data can be attributed to human error. Now, as for experiment 2, it was seen that all of the sugar test tubes had undergone fermentation, while the control test tube had not undergone fermentation. After 12 to 24 hours, the test tubes had been checked again to notice a color change and gas reduction to be able to deduce whether fermentation had occurred. Glucose, fructose, and mannitol had changed colors from red to yellow due to acid production in the phenol red broth. It was noticed that mannitol and glucose produced the most gas, while fructose produced only some, and the control did not produce any gas. And the graph for cellular respiration is shown, and you can see that the trend for the germinated seeds is higher than that of the non-germinated seeds.
While experiment 1 conclusion is as seeds were germinated, its cellular respiration rate increased, while the non-germinated seeds are dormant and use very little respiration, but still some respiration has to happen for them to continue to live. Experiment 2 taught us the volume of gas that is collected is an indication of fermentation. With more gas collected, more fermentation happening. Along with this, the color change indicates fermentation. Inherently, the production of acid due to phenol red broth, including turning yellow. Red to yellow mean lower pH. Fermentation produces acid when in the presence of sugars. During this lab, sources of human error are found, such as the seeds not being germinated properly, inaccurate readings, or the loss of gas. So ultimately, the purpose of this lab was to be able to measure aerobic respiration rates of germinated and dormant millet seeds for experiment 1, and the purpose of experiment 2 was to be able to examine yeast fermentation under experimental conditions. The purpose of this lab was ultimately achieved as seen by obtaining results that specified a trend in germinated and non-germinated seeds for the first part of this experiment, and obtaining the gas production, the color change, and the conclusion for experiment 2 for all the sugars and the control. Thank you once again to the CURE grant for going ahead and giving us this opportunity to do this project and being able to learn about cellular respiration and metabolism.